Imagine a world where the noise never truly stops. Even in the quietest room of your home, a subtle hum persists. The distant rumble of traffic, the low vibration of household appliances, the soft buzz of electronics, and the constant digital pings from our devices. For most of us, absolute silence has become a rare luxury. We have conditioned ourselves to believe that silence is simply a void, an empty space waiting to be filled. We assume that when the world goes quiet, our brain simply slips into neutral, idling like a car engine at a red light. But what if the latest discoveries in neuroscience show that the exact opposite is true? In the year 2010, a neurophysiologist named Michael Ware made a startling discovery at the University of Oregon. He found that our brains possess a highly specialized, dedicated network of neurons in the auditory cortex that do not fire when a sound is playing. Instead, these neurons ignite with electrical activity the exact millisecond a sound stops. To your brain, the sudden onset of silence is not the absence of input. It is a profound, highly active neurological event. And when that silence is sustained for just 10 minutes a day, it triggers a cascade of biological rebuilding deep inside your skull. To understand why silence is so restorative, we must first look at the biological tax that noise inflicts on our bodies every single second. Sound travels through the air as physical pressure waves. We measure this pressure on a logarithmic scale of decibels in relation to the quietest sound a human ear can detect. When these mechanical waves enter your ear canal, they vibrate your tympanic membrane, sending electrical signals along your auditory nerve directly to the amygdala, which is the brain's threat detection center. Because our evolutionary ancestors had to survive in wild, unpredictable environments, your amygdala is constantly scanning your auditory feed for signs of danger. In our modern world, even when you think you have successfully tuned out background noise, your amygdala is still actively processing those waves. It interprets this chronic, low-level background noise as a persistent environmental threat. This trigger activates your hypothalamus, which immediately kicks off a stress response. This pathway, known as the HPA axis, prompts your adrenal glands to flood your system with adrenaline and cortisol. While cortisol is helpful, in short bursts, chronic exposure to noise keeps your HPA axis active. This leads to persistent high blood pressure, arterial stiffness, sleep fragmentation, and systemic inflammation. In your brain, prolonged high cortisol levels destroy synaptic connections, accelerate memory loss, and significantly elevate the risk of developing neurodegenerative conditions like Alzheimer's disease. But the story changes completely when we step into absolute silence. For decades, scientists believed that the adult brain was structurally fixed and unable to grow new brain cells. We now know this is false. We have discovered a process called adult neurogenesis, which is the birth and functional integration of completely new neurons in a deeply nestled structure known as the hippocampus. The hippocampus is the master gatekeeper of your learning, memory, and emotional regulation. In 2013, a team of regenerative biologists led by Imke Kirste at Duke University conducted a landmark study on how different acoustic environments affect this brain region. They exposed different groups of mice to white noise, social calls, classical music, or absolute silence. To track the development of the brain cells, they used a chemical compound called BRDU, which integrates into the DNA of actively replicating cells. Initially, almost all the auditory stimuli increased the birth of new cells. But the real test of neurogenesis is whether these cells can survive and mature. When the researchers analyzed the brain 7 days later, they made an astonishing discovery. In the groups exposed to music, social calls, and noise, almost all of the newly born cells had died. Only the group exposed to 2 hours of daily silence maintained a massive, statistically significant population of surviving cells. These cells successfully matured into functional, active neurons and integrated seamlessly into the brain's existing memory networks. Why would the complete absence of sound be the only condition that actually grows and preserves brain cells? Researcher Imke Kirste formulated an elegant evolutionary hypothesis. In the wild, absolute silence is highly unnatural. When a natural habitat suddenly goes completely silent, it is an alarm signal that a predator is near. Therefore, your brain interprets absolute silence not as a state of passive rest, but as a cue requiring heightened alertness. To prepare for this unpredictable threat, the brain floods the hippocampus with fresh, highly plastic neural resources to help the animal learn, adapt, and survive. When you deliberately choose to enter a quiet space, you hijack this evolutionary survival mechanism, channeling this surge of new cells toward cognitive enhancement and emotional resilience. This physical rebuilding of the brain is supported by a profound shift in your neurochemistry. Under constant noise and digital stimulation, your dopamine pathways are over-engaged, leading your receptors to lose their sensitivity. Silence acts as a metabolic reset switch. By removing external stimulation, you allow your dopamine receptors to restore their baseline sensitivity, which brings back your intrinsic motivation and mental focus. At the same time, silence increases your serotonin production. Serotonin is the primary molecule responsible for your mood stability and sleep quality. Decreasing external noise calms the rapid firing of your serotonergic neurons, allowing your brain to accumulate and store this vital neurotransmitter. Finally, quiet environments stimulate the release of endorphins. These natural chemicals act as your body's self-synthesized pain relievers, directly reducing muscle tension and creating a deep sensation of physical safety. On a larger scale, silence alters the traffic patterns across your entire brain. Your brain constantly navigates a balance between two major, competing networks. When you are actively focused on a task, your task-positive network is in control. But the moment you stop performing external tasks and close your eyes, your brain switches to its default setting, known as the default mode network. Discovered in 2001 by neurologist Marcus Reichel, the default mode network is highly active during self-reflection, daydreaming, and memory retrieval. Under chronic stress, however, this network becomes hyperactive and poorly regulated. This is when you fall into pathological rumination, which is an exhausting, looping cycle of negative self-talk, regret, and anxiety. To calm this network, you can employ simple, structured cognitive strategies. Writing down a concrete list of action items helps your prefrontal cortex feel in control, letting the default network rest. Actively filtering what you can change from what you cannot change prevents you from wasting mental energy. Putting your problems into perspective by remembering past challenges helps reduce the perceived threat. And grounding your senses by focusing on your immediate physical surroundings pulls your brain out of its anxious mental loops. The systemic benefits of silence also extend directly to your heart and blood vessels. In 2005, cardiologist Luciano Bernardi published a landmark study in the journal Heart, measuring how different styles of music affect the human cardiorespiratory system. He found that fast-tempo music increased heart rate, blood pressure, and breathing rate, while slow music induced mild relaxation. But the most remarkable finding occurred during the randomly inserted 2-minute silent pauses. When the music suddenly stopped, the participants experienced a profound cardiorespiratory downshift. Their heart rate, blood pressure, and breathing rates dropped to levels that were significantly lower than their starting baseline. Dr. Bernardi explained that when you are listening to any sound, your brain must maintain a degree of active focus, keeping your sympathetic nervous system partially engaged. When the sound is suddenly removed, your brain experiences a release of that attention. This sudden transition allows your parasympathetic brake, which is driven by the vagus nerve, to exert maximum influence. The vagus nerve releases a chemical called acetylcholine directly onto your heart, slowing your pulse, relaxing your blood vessels, and lowering your blood pressure. This autonomic shift restores your heart rate variability, which is the key indicator of a healthy, resilient nervous system. To understand how silence restores your mental focus, we can look to attention restoration theory, developed by environmental psychologists Stephen and Rachel Kaplan. This theory explains that our ability to maintain directed, conscious focus is a finite resource managed by the prefrontal cortex. Constant distractions exhaust this resource, leading to mental fatigue, irritability, and poor concentration. The Kaplans discovered that our focus recovers when we spend time in environments that engage our involuntary attention, a state known as soft fascination. Soft fascination is effortlessly captured by gentle, natural movements, like watching clouds drift, leaves move in the wind, or water flow over rocks. Because these natural settings do not demand active decisions or analytical thinking, they allow your prefrontal cortex to completely stand down and replenish its energy. In a prominent study led by Mark Berman, participants who took a short walk through a quiet, natural setting showed a 20% improvement in working memory and focus, while those who walked in noisy urban settings showed no improvement. You do not need to attend expensive retreats or sit in silence for hours to experience these benefits. Your nervous system is highly plastic, meaning it rehearses and strengthens whatever patterns you repeat. Practicing a simple 10-minute somatosensory protocol each day is enough to recalibrate your system. First, practice active sensory deprivation. Find a quiet room, dim the lights, and turn off all digital notifications. Sit comfortably with your eyes closed. Second, engage in interoceptive grounding. Instead of letting your mind wander into anxious thoughts, actively focus on your physical sensations. Notice the weight of your body on the chair, the feeling of your feet touching the floor, or the gentle movement of your chest as you breathe. This simple shift in attention activates your insular cortex, pulling neural energy away from the overactive, ruminative areas of your brain. Third, perform exhale-emphasized breathing using a technique called the physiological sigh. Take a deep, full breath in through your nose, immediately follow it with a second, quick inhale to fully expand your lungs, and then let out a slow, extended exhale through your mouth. Make sure your exhale is twice as long as your inhale. Biophysically, when you inhale, your diaphragm moves down, temporarily shrinking your chest cavity and increasing pressure on your heart. This pressure causes a brief drop in blood flow, prompting your brain to temporarily speed up your heart rate. When you exhale, your diaphragm moves up. This expansion is detected by sensory receptors, which immediately signal your vagus nerve to slow your heart rate down. By deliberately making your exhales long and slow, you maximize this vagal activation, slowing your cardiac rhythm, lowering your blood pressure, and signaling absolute safety to your brain. Our world will likely only continue to get louder and more demanding, but by carving out just 10 minutes of deliberate silence each day, you give your brain the quiet space it needs to rebuild, restore, and protect itself.